So hello, everybody. Welcome to another edition of Meet the First Author. My name is Ron Kwon. I'm an associate professor in the Department of Orthopedics at the University of Washington. And I'm very pleased to be here with Dr. Francesca Tonelli from the Department of Molecular Medicine at the University of Pavia. Francesca, welcome. So we're here to talk about your fantastic paper that just came out in Matrix Biology. It's called CRTAP and P3H1 knockout zebrafish support defective collagen chaperoning as the cause of their osteogenesis imperfecta phenotype. So before we begin, I just wanted to say thank you for taking the time to talk to us. So as many of us know, Pavia is in the Lombardy region in northern Italy, which is one of the epicenters of the COVID-19 pandemic. So I just wanted to say thank you. And yeah, I was wondering if you could actually just tell us a little bit about what the situation is like.
Yeah, sure. So first of all, I would like to thank you to inviting me on this interview. And yes, this is a quite difficult period in Italy, and in particular in Lombardia, which is the region in which I live. Yeah, the country is locked down and all the activities has been suspended except the one that are really necessary to guarantee food and welfare. The university is open just to guarantee animal welfare and to allow technicians to reach the animal facilities. But all the students, PhD students, postdocs, researchers have to stay at home. So, of course, the laboratory, the experimental procedures are stopped, and we can just write and study and stop.
Well, thanks for the update. And how are you doing?
Yeah, we are fine. Thanks God. We are fine, and in Lombardia in the last days, it seems that the number of positive people to COVID-19 is decreasing. So we hope that in a few days we can start to diminish these infections, the rate of infections. Yeah.
Yeah, that's great, and I'm sure we're all looking forward to being able to restart our science and do the things that we love to do. So, with that being said, I'm hoping that we can sort of talk about your paper because I was really excited to see it come out. So. Could you first sort of talk about a little bit about what your lab does?
Yeah, so sure. My laboratory is focused on the study on skeletal disease and in particular osteogenesis imperfecta, which is a skeletal hereditable rare disease. And patients are characterized by bone fractures, bone fragility, and short stature. This disease can be due to mutations in genes encoding for collagen type 1 alpha chains, alpha 1 and alpha 2, but often it is caused by mutations in genes that are related to, that code for proteins involving collagen type 1, post-translational modifications, processing, and so on. So our goal is to generate animal models for this disease, both mice model, and in the last years we focus our attention on zebrafish. And yeah, we studied zebrafish since 5 years. And yeah, first we characterize a dominant form of osteogenesis imperfecta, and now we focus our attention on the generation of models using CRISPR-Cas9.
Yeah, so actually you have the honor of being actually the first author featured on this Meet the First Author feature, where you actually published a paper using zebrafish. So for those people in the community that are unfamiliar with zebrafish, can you talk a little bit about why you're using them?
Yeah. So yeah, zebrafish have several advantages because they are really small, and you can easily manipulate them because the genome is completely known, and embryos and larvae are transparent, so you can follow bone development in vivo, and it's really interesting. It is not possible using mice. and also to perform drug screening, because the final purpose of a lot of research is to perform drug screening, is possible using high number of samples, reducing time and cost. So I think that is a useful animal model to use also for studying skeletal disease.
Yeah, I think that's great. I think these are really important studies. Like you said, your lab helped develop some dominant models of OI, and now you're looking at recessive models. So can you tell us a little bit about what you did for this paper?
Yeah, so the paper was focused on the first generation of 2 zebrafish models for osteogenesis imperfecta type 7 and type 8, which are caused by mutations in CRKP, which is called also cartilage-associated protein, and the prolyl-hydroxylase 1. And this protein form a complex that is located in the endoplasmic reticulum, and help collagen to be hydroxylated, properly hydroxylated in some specific prolyl residue. But the role of this complex as also chaperone was elusive since now. So we decided to generate these knockout zebrafish models for CRKP and the prolyl-hydroxylase 1 to understand how this complex can work not only as hydroxylation for proline, but also as chaperone. And yeah. So this was the focus of our work.
And what did you guys find?
Yeah, so our zebrafish models recapitulate almost exactly the skeletal phenotype observed in osteogenesis imperfecta patients. So they are characterized by skeletal deformities, a delay in mineralization since the first weeks post-fertilization. And they have overmodified collagen type 1, which is one of the most important features that were observed also in white patients and in urine model. And we observe an interesting thing. So in mice and in patients, the prolyl-hydroxylase complex hydroxylate proline in position 986 of collagen type 1, while zebrafish, the wild-type zebrafish, has no hydroxylation of this proline. So it means that if our fish have a skeletal phenotype, this skeletal phenotype is probably due to a malfunctioning in the chaperone activity of the complex, except for the hydroxylation activity, because it is not present in the normal condition in zebrafish. So I think that this is the real important finding of this work.
Yeah, so actually in this case, you're exploiting differences in zebrafish skeletal biology to make insights into the human disease.
Yeah. Yeah.
Yeah, and I thought it was just a really beautiful paper that incorporated a number of different nice characterizations, including biochemistry and really nice quantitative phenotyping. So I just wanted to congratulate you on the work. So you're a postdoctoral fellow in Antonella Forlino's lab. What's on the horizon for you career-wise?
Yeah, so up to now I start to generate a new zebrafish model using CRISPR-Cas9, and I'm working also on both on skeletals and also some different kind of disease. I'm just right now studying ciliopathies, and yeah, I think that is a really interesting field. So yeah, we have some works that I perform also on our mutant model generated by CRISPR-Cas for the paper. That we want to try to use it as potential sample to treat with potential drugs and target therapies.
Yeah, and it sounds really exciting, and it sounds like this paper really set some strong foundations for some neat future directions. So I'm excited to see what comes out of your lab. So
thank you.
Well, thanks again for taking the time again, and yeah, take care, and thanks again.
Thanks. Thank you. Bye.
Okay, bye.